I'm delighted to be joined by Dr. Eric Jager, who's Associate Professor of Microbiology at the Center for Biopharmaceutical Education and Training in Albany, New York. Now, Eric's research focuses on enveloped RNA viruses like SARS-CoV-2, which causes COVID-19. But one misinformation campaign around these vaccines has been that they can somehow change our DNA. I suppose it's the basis of having a nucleic acid in the name. People are wondering what's the relation there. Could you outline that for us, Eric?
Certainly. You're right, where I think there's a lot of excitement around these vaccines, and they are newer to the realm of human vaccines on a wider scale. And for the audience, the way that these work, as you mentioned, is messenger RNA is kind of the blueprint that even our own bodies produce to produce proteins so that we have functions in our cells. So the idea here is that we can deliver the messenger RNA, and this provides the blueprint for the coronavirus spike protein. Our body makes that protein, and then that protein is released to educate and train our immune system to respond to the actual infection if we were to unfortunately become ill with it, so we're protected. So as you mentioned, the concern of these is people hear nucleic acids. Is that some kind of related to genetically modified organisms, GMO, and it brings in kind of that realm of discussion. Really, this type of vaccine cannot incorporate into our genome. So it's a unidirectional process. In our bodies, we have DNA, our genome creates the messenger RNA as a blueprint for proteins, and then proteins are produced. Our body does not contain machinery to go in the reverse from RNA into DNA to be incorporated into our cells. So there's safety there. Messenger RNA also is short-lived in our bodies. So once it's delivered, it does its job, it produces proteins, and then is removed. And that's important because, as I mentioned, messenger RNA is produced in our cells naturally. We're producing proteins all the time in our cells to function, and messenger RNA is removed in our cells on a daily basis and replaced by new copies. Additionally, as you mentioned, it is a new vaccine that people like, where does this come from? Is this a completely new technology I've never heard about before? But really, this vaccine and this type of vaccine, it's been studied for at least a decade or more. So this is, again, the first vaccine potentially to be used widely in the population that has a messenger RNA platform. But the use of messenger RNA in both animal studies and human studies has been going on for quite some time. So we do have scientists and clinicians do have a sense of how these vaccines work and their safety in individuals.
Great, thank you. I think a lot of people have read about the progress of these vaccines and thought, wow, that's happened so quickly, but so many other vaccines seem to take longer. But could you please elaborate more on maybe why that somehow year period is actually a bit longer than it might seem?
Right, certainly. I agree. It is kind of an interesting point is that we want a vaccine yesterday, but then we get it, and it's there's a lot of alarm about was this process rushed. And I think there's a lot of information out there that really does alleviate some concerns about that process. So the first part is that both the United States and the UK and across the globe, there are regulatory bodies that ensure that vaccines, as they go through the testing process, there's a rigorous process to make sure that they are safe. In the US here, we have different phase trials, and in both the messenger RNA and the adenovirus-based vaccines that seem the closest, they've gone through the necessary safety trials, both our country and the UK, to make sure that they're not going to have any harmful effect on individuals. And then they went into larger trials testing vaccines in 30, 40,000 plus individuals. So we have a large body of data. And I know some people say, well, is 30,000 people enough compared to the entire population of the globe? But it is a substantial number of people that they've tested the vaccine. It's been in different age groups, genders, racial groups, ethnic groups, to ensure that any individual that is going to take this vaccine should not be harmed by it. Because, of course, the idea here is do no harm. We want to give a vaccine. We're giving it to healthy people, of course. You don't want to do harm to a healthy person. So the idea here is it needs to be a benefit to the person. So there's care taken to ensure that there is no ill effects. I think additional to that is what made this process more rapid, on top of the urgency, of course, was some government programs like Operation Warp Speed in the U.S. and COVAX in the European Union to ensure that there were resources available to get this process moving along quicker. And there was also a large number of companies engaged in producing a vaccine. So I think, as you mentioned, kind of the marvel of it all, of having scientists and clinicians and epidemiologists around the world kind of connect together to make these vaccines in a quicker process. But through that, it was done safely. I think the other thing that individuals need to keep in mind in terms of the speed is that because there was such an urgency, that volunteers really stepped forward and to generate these clinical data and clinical trials usually takes a long time because it takes time to enroll subjects. It's not often something that happens very easily. But here there are people basically lining up ready to be part of this process to ensure the vaccine is safe. And the other thing too is these companies have been very transparent in the process for how their vaccines were to be tested. And they've been forthcoming with the data. The efficacy data has still been science by press release, which can be frustrating to the scientists at times, but the data, they do release it, and we do have confidence that there is, it has been safe in what we've tested. Another point I should mention, the FDA required additional safety information for the vaccine. So after individuals got the second dose, they wanted to collect 2 months of data because historically, individuals, if they're going to experience an ill effect of the vaccine, it typically happens within the first 6 to 8 weeks after an individual gets a vaccine, when the immune response is still going. So they haven't seen any serious adverse effects in those individuals given the vaccine. So short-term effects were clear that the vaccine is safe to use. There's still, caveat that we still need to collect data. There'll be safety data collected as these vaccines are rolled out broadly across the globe. There are a number of agencies that their job is to monitor on a daily basis. There's no adverse effects. And if there were, certainly the companies would pull back and try to study that. But just so people know that it's not just a end of the story after the release, people just pack up and don't think about the safety. It's something that is a concern to every scientist and every company producing these vaccines.